The overall goal of the following experiment is to observe and analyze a synchronized population of cells that have not been unduly manipulated and are still functioning normally. The position of cells within cell cycle stages is commonly determined by quantitating the DNA content by flow cytometry. To measure the time taken to transit through the cell cycle, cells are usually synchronized using conditions that block cell cycle progression, resulting in accumulation in a particular cell cycle phase. Unfortunately, this often has unintended consequences, including cell death, making results unreliable. This problem can be overcome by incubating the cells with a pulse of BrdU, a thymidine analog, to label cells in S phase. The labeled cells can be tracked through the cell cycle by harvesting at selected times after the removal of BrdU. Results obtained show the DNA content as a marker of cell cycle status for the whole population and for the cells that were in S phase during the BrdU pulse. This latter population functions as a synchronized population that can be tracked during the remainder of the cell cycle and into the next round of cell division.
The main advantage of this technique over existing methods, such as chemical blockade, is that it does not disrupt the natural cycling of the cells and limits the amount of cell death in the synchronized population.
This protocol uses the acute lymphoblastic leukemia cell line NALM6, but can be applied to any non-adherent cell line. Maintain the NALM6 cells in T75 culture flasks in complete RPMI at 37 degrees Celsius in 5% carbon dioxide in air. After collecting the cells and performing a cell count, resuspend the cells in complete RPMI at 2 × 10⁶ cells per milliliter.